Okay, let's dive in. We usually think of hardcore math calculus, complex models as being, stuck in physics labs or Wall Street trading floors.
Right, not exactly everyday stuff for most people.
But what if I told you some pretty sophisticated math is quietly shaping everything from, say, how diseases spread to predicting the exact moment your food might go bad?
It sounds a bit surprising, doesn't it?
Yeah.
But yeah, you're absolutely right. These mathematical models, they're kind of woven into the fabric of countless systems we interact with daily, often in ways we just don't see.
And that's really our mission in this deep dive, to uncover the surprising power of these tools, specifically things like integral calculus and predictive modeling,
Uh-huh.
and see how they help us understand complex dynamic systems in fields you might not immediately connect with heavy-duty calculus, like biology and social sciences.
Exactly. And we've looked at sources that explore these broad applications, the general ideas.
Hmm.
But then we're going to drill down into a really fascinating specific example,
Wait.
predictive microbiology, and its role in food safety. It's quite practical.
Okay, so we're going from fundamental math concepts like integrals all the way down to the microscopic world of, well, food spoilage.
That's the plan. It shows the range quite nicely.
Right, let's start with the fundamentals then. The integral. Maybe forget the whole area under a curve thing most of us remember from school for just a second. In this context, tracking change, what does an integral really represent?
Well, at its core, it's about accumulation. Think of it like this. If you know the rate at which something is changing at any given moment,
Like how fast a population is growing right now.
exactly, or how quickly a disease is spreading, an integral lets you sum up all those continuous, tiny, instantaneous changes over a period of time, or even across space.
Ah, so it lets you figure out the total change that happened, or the total amount of something.
Precisely. It's the mathematical way to go from knowing how fast to knowing how much total over an interval. It bridges that gap.
Okay, I think I get the concept. Accumulating change. So
Mhm.
how does this play out in biology? Because biology is just full of things changing all the time.
It really is. Population dynamics is a perfect example. If we can model the rate of population growth, maybe using a function like, say,
Right.
GT, just as an example,
Okay.
then integrating that rate function over a specific time period allows us to calculate the total increase in the population size during that time.
Or even predict the total size at some future point.
Yes, exactly.
Yeah.
It helps us understand how populations shift under different influences like birth rates, death rates, resource availability, all those factors acting continuously.
Which is incredibly useful for conservation, wildlife management, things like that.
Absolutely. It
Yep.
quantifies the overall impact, and disease spread modeling is another big one.
You mean the models public health folks use during outbreaks, like the SIR model?
Yes,
Yeah.
exactly like the SIR models, susceptible, infected, recovered.
Hmm.
Those models describe the rates at which people move between those categories.
Right.
Solving the differential equations in those models often involves integration. That's how you figure out the total number of people in the infected category, for instance, over the course of an epidemic.
So knowing the infection rate helps predict the peak of the outbreak, or how long it might last.
Precisely. It's crucial for planning responses. And this idea extends to ecology, too. Think about predator-prey relationships.
Like the Lotka-Volterra equations.
You got it. Integrating those helps accumulate the effects of their interactions over time, giving you insights into population cycles and ecosystem stability. It sums up the continuous back and forth.
Okay, so it's about taking these ongoing complex interactions and summing up their effects to see the bigger picture over time. What about social sciences then? Economics isn't quite biology, is it?
Not in the same way, no, but economies are incredibly dynamic systems. You've got prices, demand, production, all constantly fluctuating. Integrals help us quantify total outcomes that arise from these continuous changes.
Can you give an example?
Sure. Consumer surplus is a classic one. It's basically the difference between what consumers are willing to pay for something and the price they actually pay in the market.
Sort of the total extra value they feel they get.
Exactly, like that feeling of getting a good deal, but measured across the whole market.
Yeah.
If we have a demand curve, say a simple one like BPP, the BP, calculating that total surplus involves integrating.
How?
You integrate the demand function over the relevant range of quantities sold, essentially finding the area between the demand curve and the market price. It quantifies that total extra value.
That's fascinating. Using calculus to put a number on, well, collective consumer satisfaction.
It is pretty neat.
Mm.
And on a bigger scale, think about GDP, gross domestic product. Calculating the total economic output for, say, a whole year can involve integrating functions that represent income or spending over that entire time period.
Like summing up all the transactions happening every second.
Sort of, yeah. It's how economists take continuous economic activity and arrive at a total figure for the year, like integrating from day 0 to 365.
So whether it's tracking the total number of recovered patients or the total value consumers get, integrals are key for adding up continuous change to get the big picture.
They really are fundamental tools for understanding the cumulative effect of dynamic processes.
Okay, now let's transition. We've seen how these math tools work broadly. Let's apply this idea of modeling dynamic systems to a very specific area, maybe less glamorous, but absolutely critical: food safety.
Right. This brings us squarely into predictive microbiology. And it's pretty much what it sounds like, using mathematical models, often built into software, to predict how microorganisms in food will behave.
The ones that cause spoilage or even make us sick.
Exactly those. The models try to predict if they'll grow, if they'll survive processing, or if they'll be inactivated by something like heat or a preservative under different conditions.
And why is this so important, practically speaking?
Well, it directly impacts food safety, obviously, but also food quality and shelf life by predicting how microbes might behave. The food industry can make much better decisions about processing, packaging, storage temperatures, transport, everything needed to keep food safe and stop it from spoiling too quickly. It's a key part of figuring out that use-by date.
And this isn't a totally new idea in food science, is it?
Oh, not at all. Simple math models have been used for ages. Think about canning foods. The heating times and temperatures are calculated based on how fast heat kills specific microbes.
Like calculating d-values.
Precisely. The d-value tells you the time needed at a specific temperature to kill 90% of a particular microbe population. That's based on first-order kinetics, a basic mathematical model of inactivation.
Okay, so the modern stuff builds on those foundations. Let's look at how these predictive models are structured. Our sources mentioned a kind of helpful three-tier system.
Right. The Whiting and Buchanan classification is often used. At the base, you have what are called primary models.
Okay, primary. What do they do?
They focus on describing how a microbial population changes over time, but only when the environmental conditions like temperature, pH, saltiness are kept constant.
So like watching bacteria grow in a petri dish in a lab incubator where everything's held steady.
Exactly like that. You collect growth data in that controlled environment, and the primary model fits that data. It gives you key parameters like the specific growth rate, the lag phase duration, that's the initial period before growth takes off, and the maximum population density, or MPD.
Got it. Primary models track growth over time in one specific unchanging environment. What's the next level up?
That would be the secondary models.
Yeah.
These take the parameters you got from the primary models, like that growth rate or lag time, and describe how those parameters themselves change when you start varying the environmental conditions.
Ah, so this level tells you how changing the temperature or the acidity or the water activity affects how fast the microbes grow or how long they wait before growing.
Exactly. They model the relationship between the environment and the growth parameters. A simple example is the square root model for temperature's effect on growth rate. It basically says the square root of the growth rate is linearly related to temperature within a certain range.
Like the E0 BTD0.
That's the one. Of course, when you have multiple factors changing temperature and pH and water activity, the secondary models get more complex. They might use techniques like response surface analysis to map out those interactions.
And presumably, the more data you feed them from experiments under lots of different conditions, the better these secondary models get at predicting.
Absolutely. They're built on extensive experimental data sets. They improve as more knowledge is gathered.
Okay, so primary, time change in one condition. Secondary, how parameters change with conditions. What's the top tier?
Tertiary models. Think of these as the user-friendly software packages or online tools. They integrate the information from the primary and secondary models to make predictions for specific, often complex, real-world scenarios.
Scenarios where conditions might even change over time, like food cooling down or warming up.
Precisely. You might input the food type, its starting microbial load, its pH, its water activity, and maybe its expected temperature profile during storage or distribution.
And the software uses the underlying primary and secondary models to predict what will happen.
Yes. It draws on databases derived from those models to predict, say, the growth curve of Listeria monocytogenes in refrigerated deli meat, or how long it might take for salmonella to reach a certain level in eggs stored at room temperature, or even the time until toxin production by Clostridium botulinum.
So this whole tiered system effectively translates fundamental lab data on how microbes behave into practical, usable predictions for the complex world of food.
It's a really powerful framework. It brings together microbiology, mathematics, and computer science to tackle very real problems in food safety and quality.
Let's make this even more concrete. Let's apply this to a very dynamic, everyday example, the spoilage of meat. It seems like a perfect case study.
It really is.
Yeah.
Meat spoils relatively quickly because, well, it's incredibly nutritious for microbes. High water content, a moderate pH usually between 5.5 and 5.9.
And after slaughter, the animal's natural defenses are gone, right?
Exactly. Plus, post-slaughter biochemical changes occur, like the breakdown of energy reserves, the production of lactic acid, which drops the pH, and these actually make the muscle tissue even more susceptible to microbial growth.
And microbes can get onto the meat at so many points during slaughter, from equipment, handling, the environment.
Absolutely. And then storage conditions, temperature, packaging, atmosphere, humidity all play a massive role in which microbes end up dominating and causing spoilage.
It's interesting how different conditions lead to different kinds of spoilage. Can you give some examples?
Sure. So if meat is exposed to air, aerobic conditions, you often get spoilage dominated by bacteria like Pseudomonas. They can form a surface slime.
Ugh, slime.
Yeah, not appealing. Or cause off odors, sometimes color changes, a reddish bloom from some lactobacilli, or even, less commonly, blue spots from Pseudomonas cincinnati. Rancidity, too, can be caused by some Pseudomonas.
Okay, that's with oxygen. What about without oxygen, like in vacuum-packed meat?
Right. Anaerobic conditions favor different microbes. You might get souring, often caused by lactic acid bacteria, LAB, or you could get actual putrefaction with really foul smells caused by bacteria like Clostridium or Proteus.
So the packaging makes a huge difference to which microbes cause the problem.
A massive difference. In vacuum-packed meat specifically, you might get hydrogen sulfide production, that rotten egg smell from Schoenella or Hafnius species, or sometimes a greening effect.
It really sounds like a microbial battlefield where the environment, oxygen, temperature, pH dictates who wins and causes the spoilage.
That's a great way to put it. And temperature's huge. Refrigeration, say below 5 degrees C, slows most things down, but it favors cold-loving microbes, the psychrophiles.
Like Pseudomonas again.
Yes, Pseudomonas are major players in refrigerated spoilage, along with others like Brophythox thermosfacta. And worryingly, some pathogens can still grow slowly, even if it's very slowly, at refrigeration temps. Listeria monocytogenes is the big concern there, and sometimes Yersinia enterocolitica.
So even cold storage isn't a guarantee.
Not indefinitely, no. And even cured meats like hams or sausages, which have preservatives like salt and nitrate, they still spoil eventually. Different microbes cause different problems, maybe slime, maybe souring, maybe specific color defects like green patches inside caused by certain Labor or Enterococci.
You mentioned bone taint earlier.
Ah yes, bone taint. That's spoilage deep inside the meat, near the bone, often in cured hams. It's caused by bacteria like Clostridium, Serratia, or Proteus that manage to survive curing and grow in those deeper, often less salty, less acidic conditions. Gives off foul odors.
Sounds truly unpleasant. So bringing this back to the models, predicting shelf life for meat isn't just setting a timer.
Not at all. It's about using predictive microbiology models to understand this complex microbial ecosystem. Yeah. The models integrate information about the initial microbial load, the specific meat properties, pH, water activity, the packaging type, aerobic, vacuum-packed, the storage temperature profile.
And they predict when the spoilage organisms, like Pseudomonas or LEB, will reach levels that cause unacceptable changes, slime, off odors, color issues.
Exactly. Or, critically, when potential pathogens like Listeria might reach dangerous levels. The models estimate that microbial shelf life based on all those interacting factors. They translate the complex biology and chemistry into a practical prediction of safety and quality over time.
It really shows the power of applying these mathematical tools to a dynamic biological system.
Definitely. It's a crucial application for the food industry.
So reflecting on this whole discussion, we started with this core idea of integrals being about accumulating change.
Summing things up.
We saw how that applies broadly understanding shifts in populations, economies, and then we zoomed right into this very specific world of predictive microbiology.
Using similar modeling principles, rates, conditions, accumulation, to predict how tiny organisms behave in our food, specifically looking at meat spoilage as a dynamic process.
It really underlines how powerful these mathematical and modeling tools are, doesn't it? They help translate a theoretical understanding of rates and interactions into practical, concrete predictions for complex systems we deal with every day.
Yeah, the real-world impact is pretty significant when you think about it. Managing wildlife populations, guiding public health responses to disease outbreaks, understanding economic forces.
And, crucially, ensuring the safety and availability of our food supply by predicting spoilage and determining shelf life.
It's math making a tangible difference. And, it's important to remember, these models are tools. They're built on assumptions and data.
Right, they aren't crystal balls.
Exactly. Real-world systems are messy. Remember the U.S. Census example you mentioned earlier from the sources. A model can be incredibly powerful and accurate for a time, but reality can sometimes throw curveballs, meaning models need refining or have limitations.
But they are constantly evolving, getting better with more data, more computing power.
Absolutely. They are dynamic tools for understanding dynamic systems.
What really strikes me is that this isn't just abstract math taught in classrooms. This is math being actively used right now to make critical decisions, decisions affecting conservation, public health, and, well, literally determining if the food in your fridge is safe to eat tonight.
It's math in action, underpinning our understanding and management of the dynamic world, from the scale of economies down to the microscopic battleground on a piece of meat.
So maybe the next time you see something changing over time could be a news trend, a plant growing, or, yeah, even just checking the expiration date on your milk. Take a second. Consider the hidden math, the models that might be working behind the scenes, helping someone understand or predict that change. What other dynamic systems around you, things changing constantly, might be understood better with a bit of math?